Dear student, we are on the CEC Gurukul lectures and we are discussing about the characterization techniques to characterize the nanomaterials. We discussed the synthesis methods, we discussed the types of nanomaterials. Now we discuss the characterization techniques of the nanomaterial so that we can find out the properties of nanomaterials, and after getting the properties of nanomaterials we can use these nanomaterials for the further application in the various field like electronics, like in preparing the mechanical devices, we can use these nanomaterials in the biological applications. So we are discussing the characterization techniques. Up to now we discussed the various techniques. Now the next technique is the ion particle probe characterization techniques, which contains the various method. First is the CLS, that is the cathodoluminescence method. By using this method we can find out the characteristics emission of the nanomaterials. NMR, your most commonly used method, nuclear magnetic resonance spectroscopy, that are used for analysis of the odd number of the nuclear species. So NMR is the most commonly used method to characterize the nanomaterial, that also represent the magnetic property of the nanomaterials. And other methods are thermodynamic characterization techniques. In the thermodynamic characterization techniques, we deals with the temperature change, we deal with the effect of the heat on the nanomaterials, how the phase changes, that we can characterize by the thermodynamic characterization techniques. This technique contains the various methods. First is the TGA, that is the thermal gravimetric analysis. By using this method we can find out the properties like mass loss and temperature. And what is the variation in the mass loss versus the temperature, as we are dealing with the particles of the size 10 to the power minus 9. So we just see the effect of the loss of the mass with respect to temperature in the nano characterization by using this thermal gravimetry method. Next is DTA, differential thermal analysis, that is also used in the laboratory, that to find out the reaction heat capacity of the nanomaterial. DSC, differential scanning calorimetry, that also represent the reflection heat phase changes, how the phase changes of the nanomaterial as the temperature changes. So these are the thermochemical characterization techniques. Others are NC, that is the nanocalorimetry, that find out the latent heats of fusion. BET techniques also used to find out the surface area analysis, that is the Taylor method is used to find out the surface area. SURS method, colloidal size, specific surface area, we can find out by using the BET method. These are the various methods to characterize the nanomaterial. Now further we deal with the electron spectroscopy. Up to now we have seen the many characterization techniques where we use the light microscopes, where we use the microscope which use the commonly the light, but these light microscope cannot resolve structures closer than 200 nanometer. So how we can solve our purpose by using the electron microscope. It means 200 nanometer structure we cannot be characterized by using the light microscope. For this we require the electron microscope, which has a greater resolving power and the magnification. So by using the electron microscopy we can magnify the images up to 10,000 to 1 lakh and more, so that we can find out the detailed view of the bacteria, viruses, intercellular structures, molecules and the large atoms. So we require the electron microscope to get the better insight in the nanomaterials, and we can characterize the nanomaterials by getting the high resolving power and the magnification. Then we use the electron microscopy, that is use the electron as a source to analyze the surface or to characterize the nanomaterial. That method is the electron microscope. What is electron microscopy and what is the basic principle behind the electron microscopy is, when the beam of electron strike, as the name suggests, electron microscopy, it means we are using the beam of electron to characterize the specimen or to characterize the surface or to analyze the surface. When the beam of electron strikes the surface of the specimen and interacts with the atoms of the sample, it forms the signals by secondary scattering of electrons. Back scattered electrons are then characterized, generating by the information these X-ray that contain about the sample, that will analyze the surface topography and composition. What does it mean? That by electron microscopy we pass the electrons on the sample. When it strike on the sample, then there will be the electron and metal interaction. Electron or material interaction will take place. After that, the electrons will be ejected, that is in the form of the secondary scattering electrons, and the back electrons or electrons that will be used for the further analysis of the material that represent the surface morphology, its topography, composition and its other properties also. So this is the basic principle of the electron microscopy. On the screen you are seeing a diagram that here we are this is the commonly used method of the electron microscopy, where we use the sample. Electron beam pass from the various lenses and fall upon the sample, and through which we can find out the secondary electrons that can be characterized. So these are the schematic diagram of the electron microscopy. You are seeing here that we are having the magnetic lens. On the upper side we have the electron beam that pass from the anode, then it pass from the magnetic lenses so that it can go straight on the sample. After that it pass from the scanning coil. From the scanning coil it pass from the another lens, and from this lens it fall upon the sample, and from the sample the back scattered electrons can be detected. And between the scanning coil and the sample we are having the ring type structure, that is a detector that connect with the computer and to the scanner that will analyze the material. And from this we can find out when the electrons strike on the sample, we can get the back scattered electron, and that can be detected. Right hand side we have the secondary electron detector that detect the secondary electron generated and emitted from the surface of the material when the electron strike on it. So this is the basic structure of the electron microscopy, which uses the magnetic lenses and condensed lenses, and that is the anode, and it uses the scanner that will give the output. So this secondary electron detector connect with the computer or the scanner that will give the output. So this is a very common method. Next, here we use the energy source is a beam of electron. As a electron spectroscopy, the beam of electron is used to scan the surface. Imaging is created and viewed on the monitor. It means when the electrons strike on the sample, the output we can get on the monitor, computer or the scanner. beam of the electron used to produces the images. That beam of the electron give the surface images of the material. The wavelength of the electron beam is much shorter than the light, so that it is having a power of the magnification. So we can magnify the images and getting the high resolution power by using this. The resulting is wavelength of the electron beam is much shorter than the length, resulting in the much higher resolution by using the electron microscope. High resolution images we can get by using the electron microscope. Here we see the type of electron microscope. The two most commonly microscope are transmission electron microscope and second is the scanning electron microscope. As the name suggests, transmission electrons will be transmitted into the sample. Second is the scanning electron microscope, electron used to scan the surface. But the common method is both is using the common method, only the change of the position of the sample. So types of electron microscopy: wide field microscopy, confocal. We see on the left hand side, it is a wide field microscopy where we use the photon that light is emitted with the electron. Glass lenses are substituted with the electromagnetic and electrostatic lenses, so that a straight beam can pass through the sample, and we can get the transmission electron microscopy. The same process we use here. We use the confocal lenses scanning microscope, and that is also used the photon. Then finally we get the scanning surface analysis. Again here we also use the glass lenses, condensed lenses, object lenses, electromagnetic lenses, so that we can get the straight beam of the light which can fall upon a sample. So these are the two methods we will discuss further. SEM, scanning electron microscope is a small variation is the SEM that adds the three-dimensional viewing of the sample. By using the SEM we can find out the three-dimensional view of the sample, and we can also get the colored picture of the structure of the material. In the scanning electron microscopy, signals emitted from the surface of thick specimens. Narrow beam will be used, and it is having some physical limitations of this. The scanning electron microscope will further discussed. In the scanning electron microscope signals, it is used the surface of the thick specimen, so that we can get the clear surface analysis of the material. TEM, that is a transmission electron microscope. It utilizes the staining procedures prior to use. In transmission electron microscopy, electron pass through the thin specimen. A thin specimen is used so that light can be transmitted into the material, and the thin specimen of the size 50 to 1000 nanometer. For this we have to use the bulk beam of electrons to characterize the nanomaterial to get the electron microscopy. Next, what is the component behind the electron microscope? What are the components required in the electron microscope? The most common, common component is the electron gun. It means electron source, which is used to characterize the material which fall upon the surface and which can scan the surface. So electron source is the most important component of the electron spectroscopy. So for this light microscope, that is a tungsten filament, bright field we use, and electron microscope use a tungsten filament that is of the common form. Right? There is a difference between these two. Next, electron source. Here we see in the diagram that there is a filament is heated, electrons are emitted from the tip, that is represented by the F. F is the filament, and W is the electrode, and C is the ceramic high voltage insulator that is connected with this filament, and RB is the auto biased resistor, and IE is electron emission current. And by using this we can get the electron source. Next, what is happening here? In elastic scattering is taking place. What is the electron and specimen interaction? The primary electrons hit the electrons of the specimen atoms. Primary electrons will strike on the specimen atoms, and from there energy is transmitted from the primary electrons to the secondary to the specimen. Here the beam of electron when fall upon the material, then this energy is transferred to the specimen atoms, and that will emit the electrons and produce the radiation. When the electron strike with the specimen, then energy is transferred from the primary electron to the specimen, then emission of the electron takes place and it generates the radiation. That is the common steps of the electron specimen interaction. Here in the diagram, what we can get from the electron and specimen interaction. Here we see that when the primary electron strike with the specimen, and there is interaction takes place between the primary electron and the specimen. And what output we can get from this method? What type of outputs we can get from this method? First is, just you see from the primary electron, that is on the red line is defining here, and when it pass from the specimen, then we can get the just on the downward statement, that is a unscattered electron. It direct pass from the sample and we are getting the unscattered electrons. Now further, you see on the left hand side, then what type of output we can get and what type of electron specimen interaction. We can find out the backscattered electrons, and we can take the X-rays. The electron will be ejected. Auger electrons will be generated when the primary electrons strike with the inner surface of the specimen, and secondary electrons will be generated, and cathode luminescence we can get. That is the upper part. That represented in the blue. That is the SEM analysis we can done by the primary electrons. Here we see in the diagram that all characterizations are going to be done on the surface of the material. It means surface analysis of the specimen can be characterized by the SEM, scanning electron microscope. Like we can find out the backscattered electrons, X-rays, Auger electrons, secondary electrons, and cathode luminescence when the primary electron strikes on the surface or scan the surface. But what about when it pass from the or transmit from the specimen? Then we can get the TEM, that is the TEM analysis, transmission electron analysis. By TEM we can find out the structure of the inner surface of the specimen, as well as we can get the inelastic scattering of the electrons, elastic scattering of the electrons, that we also get from the TEM analysis. For this we use the transmission electron microscope. So this electron specimen interaction divided into two parts. First, on the upper surface area will be scanned, and second is the transmission of the primary beam primary beam from the surface to the inner structure, and then we can get the output. So both are having the different position in the apparatus arrangement. So scanning electron analysis, which scan the surface, transmission electron analysis, which analyzes the beam passed from the specimen. So these are the two most commonly methods which can find out the various properties, surface properties, as well as composition, as well as morphology of the specimen, which are of the nano size. So further, what we are seeing here, this is the image that represent, this is a very simple image that represent the, this is the surface, black line it represent the surface, from upper line it is the blue part, it representing the electron beam fall upon a surface, and from below the blue line we are having the three lines also, that is the inelastic scattering, right hand side elastic scattering, and left hand side we have the secondary electrons. On the upper side we are having the auger electrons, and we can get the cathodoluminescence, and then back scattered electrons we will get. An X-ray also we are getting by the electron and specimen interaction. This is the view of the specimen and electron interaction. Next, general characterization techniques which are used are in the electron microscopy are scanning electron microscopy, that is SEM, transmission electron microscopy, TEM, scanning tunneling microscopy. We will discuss all these techniques in detail, and these are most commonly techniques to characterize the morphology or the surface analysis or the transmission spectroscopy and the scanning tunneling microscopy of the samples which we prepared by the synthesis. So these three methods we will discuss in the detail. First method is the scanning electron microscope, that is SEM, that is used to visualize the surface of the object or the specimen, that is used to visualize the surface of the object. How we can, you are seeing on the screen there is a machine that is used on the screen, you are seeing a machine behind the text, that is the scanning electron microscope, which is used to characterize the surface of the object. So what we can see by using the SEM analysis, first is the topography. Topography means texture, surface of the sample, and morphology, in the morphology we can find out the size, shape, order of the particles. Next, composition, elemental composition of the sample we can find out by using this SEM technique. Crystalline structure also find out, like arrangement of the atoms and molecules within the sample, that we can also find out by using the SEM technique. So by using the SEM technique we can find out the topography, morphology, composition, crystalline structure. These are the basic features we can find out by using the SEM. What are the operational mode of the SEM? It works on the three modes. First is the primary mode, that use the high resolution 1 to 5 nanometer, and secondary electron imaging. Secondary part is the characterization of X-ray, identification of the element, composition of the sample by EDX techniques. Third, tertiary is the backscattering, electronic images and clues to the elemental composition of the sample. What are the elements is doped in the material, that can be found out by using the SEM techniques. What are the basic principle behind the SEM? Electronic devices are used to detect and amplify the signal, that display them as an image on the cathode ray tube, in which the raster scanning is synchronized with that of microscope. Similarly, in the similar manner as we discussed in the electron microscope, here we use the here we use the electron beam that will be used to scan the surface. So electronic devices are used to detect the amplifying the signal, and then these signals can be imaging and can be output on the microscope and on the screen we can get. In the typical SEM, the beam passes through the pair of scanning coils or pair of deflector plates in the electron column to the final lenses. The beam which is generated from the coil, here we see on the right hand side diagram that there is from the yellow part there is a filament from which electron beam is passed, and that pass from the scanning coil, and from the detector plate it finally pass from the lens and fall upon the sample, which detect the beam horizontally and vertically. This beam fall upon a sample and output we will get on the screen. So in the typical SEM, the beam, electron beam will pass from the various deflectors in the column of the final lenses, which deflect the beam horizontally and vertically so that it can fall at a particular angle on the specimen. Next, the image displayed is therefore a distribution map of the intensity of the signal being emitted from the scanned area of the specimen. What it represents? The detector will detect the image of the sample that is in the form of the structure or the signal, or it will define the scanned area of the specimen. Either we select the particular area of the specimen. So the image we can get on the screen. Here we see in the diagram, this is the schematic diagram of the SEM. On the upper part we are having the electron gun. From here electron beam generated that pass from the condensed lens. You see here the electron beam is managed accordingly, and it will pass and it will deflected at a particular angle so that it pass from the scan coil, then it pass from the objective lens in a proper manner, and then it will fall upon the specimen. From the specimen, the sample is on the lower side, and on the specimen is then the specimen attached with the detectors that will detect the backscattered scattered electrons, and from right hand side you can see the output image of the specimen. The basic principle: electron fall upon the electron beam, pass from the condensed lens, then it pass from the scan coil, then it pass from the objective lens, and at a particular angle it fall upon a specimen. From the specimen we can get the output or in the form of the scanned surface, that is in the form of the backscattered electron, scattered electron, and that can be detected by the detector. Various detectors attached with the output, and these detectors will produce the images of the surface. By getting these images we can find out the various properties of the nanomaterial. Thank you very much.